The following video gives an overview of the Alliance Separations module. This video should be viewed prior to going on site to perform the OJT on the Alliance system. There are two types of Alliance systems, the Platinum Alliance system and the Blue Alliance system, which was released in 2013. Both have the same specifications. However, the Blue Alliance has some advantages, including improved solvent and waste management, and the UV detectors on the Blue Alliance system have enhanced thermal stability. Next, let's take a look at the components of each of these systems and how they relate to each other. Here we have the detector, the solvent bottle tray, the syringe door, the sample management system, the column heater, the degasser tray, which is inline, and the solvent management tray. Now that we're familiar with what the components are in the stack, let's talk about how these components work together on this system. We will start with the path of the mobile phase, as it is a vehicle for the sample. The mobile phase travels from the bottles down to the degasser through some tubing. And after the mobile phase is degassed, it travels through the rest of the pump components. In this animation, we are showing a quaternary solvent manager, which is the type of solvent manager on the Alliance system. After degassing, the mobile phases travel through the GPV, gradient proportioning valve, the primary pump, through a transducer, through the accumulator, the other transducer, the prime vent valve, and the inline filter. The mixed solvent then travels to the sample manager, through the injection valve, and through the column, and onto the flow cell of the detector. When the sample is injected, it travels through the needle to the injection valve, then through the column where the mixture separation takes place. The separated analytes travel to the flow cell of the detector. The detector passes light through the flow cell, measures the absorbance, and processes the signal to produce a chromatogram. Now let's discuss some of the features on the 2695 Alliance system. This system has a quaternary GPV, gradient proportioning valve, as discussed in the previous slide. It has 120 sample vial capacity. Standard 2 ml vials are used with this system. It has an independent needle and plunger seal wash. It is IEEE and RS-232 compatible, Ethernet controllable. There is a column heater or column heater cooler available, a sample heater chiller available, and there is detector flexibility where detectors can be interchanged depending on the customer's preferences. There are also optional kits that come with the system, listed here. Part numbers are also listed for convenience. Other component options include syringes. So the standard syringe is 250 microliters. Other syringes that can be purchased can be 25 microliters, 100 microliters, or 2500 microliters. Also an option is different sample loop sizes. The standard sample loop size is 100 microliters. These are available in variable sizes up to 2 milliliters. The weight of the system is 130 pounds. The total system volume is less than 650 microliters. As far as other component options, the column heater with or without the valve is available. However, without the valve is default. The column heater cooler, again, with or without the valve is available, and without the valve is default. There are several field installable column switching valves that can be purchased by the customer. A 2-position regeneration 10-port valve, a 3-position selection valve, or a 6-position selection valve. Next, let's take a look at the components inside of the system. If we took off the covers and looked inside the left side of the system, we would see the CPU board shown here. Pump tray assembly is also shown down here. And in addition, you can see the cable to the CPU board shown here. If we take a look at the right-hand side without the covers, we would be able to see the injector. We would also be able to see the syringe drive, the carousel drive here, the needle pump wash shown here, and then valve 1 through 4 is also in this general area, and we'll talk more about that later. In addition, we'll be able to see the loop here, this yellow loop, which is the 100 microliter loop. We will see the degasser assembly shown here. So here we have the degasser chambers, our GPV valve down here. Next, if we look at the rear side of the instrument, we would see the old style shown here and the new style shown here. So here's the Ethernet connection. We can also see just a little bit of the heater cooler shown here. RS-232 connections and the IEEE connection if available on that system. Now let's take a look more closely at all of the components of the system. Once again, we will explore each component in the order the mobile phases travels through the system. So first, we'll take a look at the external tubing. The solvent tubing is usually colored depending on which line it is, but even if it's not colored at the customer sites, the lines should be labeled. The picture to the right shows all the lines, including the red line going to the column or column selection valve, depending on the configuration. This explains the various solvent lines and the tubing associated with them. If the solvent lines are not labeled while going to the customer site, and you must remove more than one at a time, make sure to tape them and label them so you don't lose track of which one is which. Next, the mobile phases enter the degasser tray. The purpose of the degasser is to remove any gases from the solvent. Here is the degasser tray. Here we have the GPV valve housing, degasser chambers, degasser motor, and PCB. If you were to look inside of the degasser chamber, it would look like this. This is the semi-permeable membrane that the mobile phases pass through. You do not need to replace all of the chambers if one is bad. You can swap them out to isolate the bad chamber as well. The degasser PCB controls the vacuum, and you can check the board by removing the hose from the PCB and testing the vacuum itself. Chambers should not be taken apart since you probably won't be able to get the vacuum seal back. Remember, all these chambers are connected, so if one has an internal leak after a period of time, they will all have solvent in them. The current style degasser has 4 channels, and the degasser on the 2795 Alliance system has 5 channels. As far as best practices to ensure the life of your degasser, ensure lines are never run dry. Make sure to prime all chambers and flush salts from chambers when not in use. So after the mobile phase is degassed, it goes through the rest of the solvent manager, which has a serial flow path design. Here we have a picture of the actual solvent manager. The degasser tray mentioned in the last slide is shown here. This is just a front view. If we look at the path itself, the mobile phase goes from the degasser to the GPV valve. The valve then proportions the mobile phase determined by the method and parameters, then flows to an inlet check valve, into the primary pump head, then through the transducer, another check valve to the accumulator, through the system transducer, and then through the prime and vent valve, then onto the inline filter and to the sample manager. Now let's talk a little bit more about transducers. Transducers are used throughout the instrument to measure the pressure of the system. The system pressure transducer measures the operating pressure. A software algorithm then compares the primary pump head pressure and the system pressure readings. The system then regulates the pre-compression step to balance the pressures, providing a smooth, ripple-free flow. The primary transducer senses back pressure developed by resistance to the solvent flow in the primary piston chamber